Hi, my name is Ben Cardoen. I'm a PhD student at Simon Fraser University under supervision by Professor Ghassan Hamani and Professor Ivan Robert Nabi. I'd like to present to you quickly our work in using belief theory to automatically and adaptively detect objects in super resolution and confocal microscopy. So what you see here are the two use cases we applied our method on. On the left-hand side, we see a 2D STED super resolution image of a cancer cell. Our aim here is to identify the Caveolae, which are objects that we'll discuss later. But in a brief summary, these objects are continuously being constructed and destructed in order to give a membrane a certain flexibility with respect to isotonic and mechanical stress. So at the top row you see the results, and you see the raw images, and in the lower row you see the results, where red indicates Caveolae, green indicates the building blocks or scaffolds, and orange indicates the intermediate shape. In very faint blue, you see actually identified non-specific labeling. On the right-hand side, you see the confocal microscopy of retinal tissue of an Alzheimer positive diagnosed patient. The aim here is to identify which amyloid beta deposits are actually indicative of disease and which are present in normal patients. This is a hard problem because amyloid beta is naturally occurring, so we really need to identify locally what parts are actually indicative of disease. So after that introduction, we'll focus on the Caveolae use case. So Caveolae are hemispherical, about 100 nanometers wide invaginations of the cell membrane. And you see in subpanel A the construction from all the way from Cav1 protein to more complex formations of Caveolae. The intermediate stage here, the Cav1 scaffolds, will be identified in green, and then the final Caveolae in red. There's also a non-specific binding where a fluorophore isn't actually targeting the Cav1 protein. It's been known that this actually can start to cluster and then form pseudo-objects, so it's important to identify those. Those will be marked in blue. So in order to be able to learn which objects are actually Caveolae, which are scaffolds, and so forth, we have three cell lines. One where nothing is actually altered, which is red, it's called PT3-PTOF. Green, where PTOF is disabled. PTOF is a protein that is needed for Caveolae formation. So there, in that cell line, in the green cell line, there's no Caveolae at all. And then finally, in blue, we also knock out Cav1. So there, only non-specific binding is going to be present. So with those three cell lines, we can apply contrastive learning using belief theory to actually, after the object detection, actually start identifying each object in the cell. In a very concise overview, you see in panel C the entire flow chart, so split into the object detection stage and the labeling stage. The object detection stage is designed to be fully automatic, self-tuning, and adaptive to the images, and you can see in panel D why that actually is needed. For example, the knockout images are far fainter, and the object distribution is also completely different. Our method uses the kurtosis to actually automatically compute its optimal parameters. So then after that's done, you need to compute a label that at a high level expresses like how likely is it that this object is backgrounds, scaffolds, or Caveolae. And to do that, we use belief theory, which is a superset of probability theory, and there's a small frame to actually indicate the difference, but at a high level, it means that instead of a single probability value, you get a lower or possibility value and a higher plausibility value, and then the uncertainty in between. And this enables us to actually come up with this contrastive learning method. We don't have object foreground truth, but we do know that the frequency of Caveolae in these cells using SMLM by Dr. Ishmael Gather is at 20% versus 80% scaffold, and we do see here in panel A that the probability density function actually has an inflection point at that 20% level, where left side is the probability that an object is Caveolae and right side that the objects are scaffolds. We also see that the correlation with P2F, which is required for the formation, actually increases with our label, also suggesting that what we're detecting are actually Caveolae. And you do see that if we actually do a low smoothing, that inflection point coincides with that frequency inflection point, suggesting that we're actually quite accurate. Finally, we're also capable of recording conflict. That is, if you have a model computed on different data sets, our model is actually able to tell you how much of a conflict there is between the labels that are being computed. And with that, I am open for more questions in the chat or via email. Thank you.